Hello everyone, welcome to the fourth module of the paper Cell Biology. In this module, we will study about the ultrastructure of the endoplasmic reticulum and Golgi bodies, which are some of the major cell organelles. This module would also give us a fairly good idea about the various functions of these organelles and the important roles they perform in the cell. Structure and function of endoplasmic reticulum. Eukaryotic cells are distinguished from prokaryotic cells by the presence of membrane enclosed organelles within their cytoplasm. These organelles provide discrete compartments in which specific cellular activities take place, and the resulting subdivision of the cytoplasm allows eukaryotic cells to function efficiently in spite of their large size. Ultrastructure of endoplasmic reticulum. The endoplasmic reticulum is a network of membrane enclosed tubules and sacs that extend from the nuclear membrane throughout the cytoplasm. The entire endoplasmic reticulum is enclosed by a continuous membrane and is the largest organelle of most of the eukaryotic cells. Its membrane may account for about half of all cell membranes, and the space enclosed by the endoplasmic reticulum, also called the lumen or cisternal space, may represent about 10% of the total cell volume. There are three contiguous membrane domains within the endoplasmic reticulum that perform different functions within the cell. The rough endoplasmic reticulum, which is covered by ribosomes on its outer surface, and the translational endoplasmic reticulum, where vesicles exit to the Golgi apparatus, both function in protein processing. The smooth endoplasmic reticulum is not associated with ribosomes and is involved in lipid rather than protein metabolism. Different roles of endoplasmic reticulum. The role of endoplasmic reticulum in protein processing and sorting was first demonstrated by George Pelade and his colleagues in the 1960s. These investigators studied the fate of newly synthesized proteins in specialized cells of the pancreas that secrete digestive enzymes into the small intestine. These experiments defined a pathway taken by secreted proteins, the secretory pathway, that is, rough endoplasmic reticulum to Golgi to secretory vesicles to cell exterior. The entrance of proteins into the endoplasmic reticulum thus represents a major point for the traffic of proteins within eukaryotic cells. Proteins destined for secretion or incorporation into the endoplasmic reticulum, Golgi apparatus, lysosomes or plasma membrane are initially targeted to the endoplasmic reticulum. In mammalian cells, most proteins are transferred into the endoplasmic reticulum while they are being translated on membrane bound ribosomes. In contrast, proteins destined to remain in the cytosol or to be incorporated into the nucleus, mitochondria, chloroplasts or peroxisomes are synthesized on free ribosomes and released into the cytosol when their translation is complete. Targeting proteins to the endoplasmic reticulum. Proteins can be translocated into the endoplasmic reticulum either during the synthesis on membrane bound ribosomes, which is called co-translational translocation, or after the translation has been completed on free ribosomes in the cytosol, which is called post-translational translocation. In mammalian cells, most proteins enter the endoplasmic reticulum co-translationally, whereas both co-translational and post-translational pathways are used in yeast. The first step in the co-translational pathway is the association of ribosomes with the endoplasmic reticulum. Ribosomes are targeted for binding to the endoplasmic reticulum membrane by the amino acid sequence of the polypeptide chain being synthesized, rather than by intrinsic properties of the ribosome itself. Ribosomes engaged in the synthesis of proteins that are destined for secretion are then targeted to the endoplasmic reticulum by a signal sequence at the amino terminus of the growing polypeptide chain. These signal sequences are short stretches of hydrophobic amino acids that are usually cleaved from the polypeptide chain during its transfer into the endoplasmic reticulum lumen. The general role of signal sequences in targeting proteins to the appropriate locations within the cell was first elucidated by studies of the import of secretory proteins into the endoplasmic reticulum. These experiments used in vitro preparations of rough endoplasmic reticulum, which were isolated from the cell extracts by density gradient centrifugation. When cells are disrupted, the endoplasmic reticulum breaks up into small vesicles called microsomes. Because the vesicles derived from the rough endoplasmic reticulum are covered with ribosomes, they can be separated from similar vesicles derived from the smooth endoplasmic reticulum or from other membranes, example the plasma membrane. The mechanism by which secretory proteins are targeted to the endoplasmic reticulum during the translation is now well understood. The signal sequences span about 20 amino acids, including a stretch of hydrophobic residues, usually at the amino terminus of the polypeptide chain. As they emerge from the ribosome, signal sequences are recognized and bound by a signal recognition particle called SRP, consisting of 6 polypeptides and a small cytoplasmic RNA called as 7-SL RNA. SRP binds the ribosome as well as the signal sequence, inhibiting further translation and targeting the entire complex, which includes the SRP, ribosome and the growing polypeptide chain to the rough endoplasmic reticulum by binding to the SRP receptor on the endoplasmic reticulum membrane. Binding to the receptor releases the SRP from both the ribosome and the signal sequence of the growing polypeptide chain. The ribosome then binds to a protein translocation complex in the endoplasmic reticulum membrane, and the signal sequence is inserted into a membrane channel. In both yeast and mammalian cells, the translocation channels through the endoplasmic reticulum membrane are complexes of 3 transmembrane proteins called the Sec61 proteins. The yeast and mammalian Sec61 proteins are closely related to the plasma membrane proteins that translocate secreted polypeptides in bacteria, demonstrating a striking conservation of the protein secretion machinery in prokaryotic and eukaryotic cells. Transfer of the ribosome from the SRP to the Sec61 complex allows translation to resume, and the growing polypeptide chain is transferred directly into the Sec61 channel and across the endoplasmic reticulum membrane as translation proceeds. Thus, the process of protein synthesis directly drives the transfer of growing polypeptide chains through the Sec61 channel and into the endoplasmic reticulum. As translocation proceeds, the signal sequence is cleaved by signal peptidase and the polypeptide is released into the lumen of the endoplasmic reticulum. Insertion of proteins into the endoplasmic reticulum membrane. Proteins destined for secretion or residence within the lumen of the endoplasmic reticulum, Golgi apparatus or lysosomes are translocated across the endoplasmic reticulum membrane and released into the lumen of the endoplasmic reticulum as already described. However, proteins destined for incorporation into the plasma membrane or the membranes of the endoplasmic reticulum, Golgi or lysosomes are initially inserted into the endoplasmic reticulum membrane instead of being released into the lumen. From the endoplasmic reticulum membrane, they proceed to their final destination along the same pathway as that of secretory proteins, that is, from the endoplasmic reticulum to the Golgi to the plasma membrane or lysosomes. These proteins are transported along this pathway as membrane components, however, rather than as soluble proteins. Integral membrane proteins are embedded in the membrane by hydrophobic regions that span the phospholipid bilayer. The membrane spanning portions of these proteins are usually alpha helical regions consisting of 20 to 25 hydrophobic amino acids. The formation of an alpha helix maximizes hydrogen bonding between the peptide bonds, and the hydrophobic amino acid chains interact with the fatty acid tails of the phospholipids. However, different integral membrane proteins differ in how they are inserted. For example, whereas some integral membrane proteins span the membrane only once, others have multiple membrane spanning regions. In addition, some proteins are oriented in the membrane with the amino terminus on the cytosolic side, others have carboxy terminus exposed to the cytosol. These orientations of proteins inserted into the endoplasmic reticulum, Golgi, lysosomal and plasma membranes are established as the growing polypeptide chains are translocated into the endoplasmic reticulum. The lumen of the endoplasmic reticulum is topologically equivalent to the exterior of the cell. So, the domains of plasma membrane proteins that are exposed on the cell surface correspond to the regions of polypeptide chains that are translocated into the endoplasmic reticulum. Proteins can also be anchored in the endoplasmic reticulum membrane by internal signal sequences that are not cleaved by signal peptidase. These internal signal sequences are recognized by the SRP and brought to the endoplasmic reticulum membrane as already discussed. Because they are not cleaved by signal peptidase, however, these signal sequences act as transmembrane alpha helices that exit the translocation channel and anchor proteins in the endoplasmic reticulum membrane. Importantly, internal signal sequences can be oriented so as to direct the translocation of either the amino or carboxy terminus of the polypeptide chain across the membrane. Therefore, depending on the orientation of the signal sequence, proteins inserted into the membrane by this mechanism can have either the amino or carboxy terminus exposed to the cytosol. Protein glycosylation in the endoplasmic reticulum. The covalent addition of sugars to proteins is one of the major biosynthetic functions of the endoplasmic reticulum. Most of the soluble and membrane bound proteins that are made in the endoplasmic reticulum, including those destined for transport to the Golgi apparatus, lysosomes, plasma membrane or extracellular space, are glycoproteins. In contrast, very few proteins in the cytosol are glycosylated, and those that carry a much simple sugar modification in which a single N-acetyl glucosamine group is added to a serine or threonine residue of the protein. An important advance of the understanding of the process of protein glycosylation was the discovery that a preformed precursor oligosaccharide composed of N-acetyl glucosamine, mannose and glucose and containing a total of 14 sugars is transferred en bloc to proteins in the endoplasmic reticulum. Because this oligosaccharide is transferred to the side chain amino group of an asparagine amino acid in the protein, it is said to be N-linked or asparagine linked. The transfer is catalyzed by a membrane bound enzyme, an oligosaccharyl transferase, which has its active site exposed on the luminal side of the endoplasmic reticulum membrane. This explains why cytosolic proteins are not glycosylated in this way. The precursor oligosaccharide is held in the endoplasmic reticulum membrane by a special lipid molecule called dolicol, and it is transferred to the target asparagine in a single enzymatic step immediately after that amino acid has emerged into the endoplasmic reticulum lumen during protein translocation. Since most proteins are co-translationally exported into the endoplasmic reticulum, N-linked oligosaccharides are almost always added during protein synthesis. The Golgi apparatus. The Golgi apparatus or Golgi complex functions as a factory in which proteins received from the endoplasmic reticulum are further processed and sorted for transport to the eventual destinations, lysosomes, the plasma membrane or secretion. In addition, as noted earlier, glycolipids and sphingomyelin are synthesized within the Golgi. In plant cells, the Golgi apparatus further serves as a site at which the complex polysaccharides of the cell wall are synthesized. The Golgi apparatus is thus involved in the processing the broad range of cellular constituents that travel along the secretory pathway. Organization of the Golgi. Morphologically, the Golgi is composed of flattened membrane enclosed sacs and associated vesicles. A striking feature of the Golgi apparatus is its distinct polarity in both structure and function. Proteins from the endoplasmic reticulum enter as its cis phase, which is convex and usually oriented towards nucleus. They are then transported through the Golgi and exit from its concave trans phase. As they pass through the Golgi, proteins are modified and sorted for transport to their eventual destinations within the cell. Distinct processing and sorting events appear to take place in an ordered sequence within different regions of the Golgi complex. So, the Golgi is usually considered to consist of multiple discrete compartments. Although the number of such compartments has not been established, the Golgi is most commonly viewed as consisting of 4 functionally distinct regions: the cis Golgi network, the Golgi stack, which is divided into the medial and transub compartments, and the trans Golgi network. Proteins from the endoplasmic reticulum are transported to the endoplasmic reticulum Golgi intermediate compartment and then enter the Golgi apparatus at the cis Golgi network. They then progress to the medial and trans compartments of the Golgi stack, within which most metabolic activities of the Golgi apparatus take place. The modified proteins, lipids and polysaccharides then move to the trans Golgi network, which acts as a sorting and distribution center, directing molecular traffic to lysosomes, the plasma membrane or the cell exterior. Although the Golgi apparatus was first described over 100 years ago, the mechanism by which proteins move through the Golgi apparatus has still not been established and is an area of controversy among cell biologists. One possibility is that transport vesicles carry proteins between the cisternae of the Golgi compartment. However, there is considerable experimental support for an alternative model proposing that proteins are simply carried through compartments of the Golgi within the Golgi cisternae, which gradually mature and progressively move through the Golgi in the cis to trans direction. Protein glycosylation within the Golgi. Protein processing within the Golgi involves the modification and synthesis of the carbohydrate portions of glycoproteins. One of the major aspects of this processing is the modification of the N-linked oligosaccharides that were added to proteins in the endoplasmic reticulum. As discussed earlier, proteins are modified within the endoplasmic reticulum by the addition of an oligosaccharide consisting of 14 sugar residues. 3 glucose residues and 1 mannose are then removed while the polypeptides are still in the endoplasmic reticulum. Following transport to the Golgi apparatus, the N-linked oligosaccharides of these glycoproteins are subject to extensive further modifications. N-linked oligosaccharides are processed within the Golgi apparatus in an ordered sequence of reactions. The first modification of proteins destined for secretion or for plasma membrane is the removal of 3 additional mannose residues. This is followed by the sequential addition of an N-acetyl glucosamine, the removal of 2 more mannoses and the addition of a fucose and 2 more N-acetyl glucosamines. Finally, 3 galactose and 3 sialic acid residues are added. Different glycoproteins are modified to different extents during their passage through the Golgi. Depending on both the structure of the protein and on the amount of processing enzymes that are present within the Golgi complexes of different types of cells. Consequently, proteins can emerge from the Golgi with a variety of different N-linked oligosaccharides. Lipid and polysaccharide metabolism in the Golgi. In addition to its activities in processing and sorting glycoproteins, the Golgi apparatus functions in lipid metabolism, in particular in the synthesis of glycolipids and sphingomyelin. As discussed earlier, the glycerol phospholipids, cholesterol and ceramide are synthesized in the endoplasmic reticulum. Sphingomyelin and glycolipids are then synthesized from ceramide in the Golgi apparatus. Sphingomyelin, the only non-glycerol phospholipid in cell membranes, is synthesized by the transfer of a phosphorylcholine group from phosphatidylcholine to ceramide. Alternatively, the addition of carbohydrates to ceramide can yield a variety of different glycolipids. Protein sorting and export from the Golgi apparatus. Proteins as well as lipids and polysaccharides are transported from the Golgi apparatus to their final destinations through the secretory pathway. This involves the sorting of proteins into different kinds of transport vesicles which bud from the trans-Golgi network and deliver their contents to the appropriate cellular locations. Some proteins are carried from the Golgi to the plasma membrane by a constitutive secretory pathway which accounts for the incorporation of new proteins and lipids into the plasma membrane as well as for the continuous secretion of proteins from the cell. Other proteins are transported to the cell surface by a distinct pathway of regulated secretion or are specifically targeted to other intracellular destinations such as lysosomes in animal cells or vacuoles in yeast. Proteins that function within the Golgi apparatus must be retained within that organelle rather than being transported along the secretory pathway. In contrast to the endoplasmic reticulum, all of the proteins retained within the Golgi complex are associated with the Golgi membrane rather than being soluble proteins within the lumen. The signals responsible for retention of some proteins within the Golgi have been localized to their transmembrane domains which retain proteins within the Golgi apparatus by preventing them from being packaged in the transport vesicles that leave the trans-Golgi network. In addition, like the KKXX sequences of the resident endoplasmic reticulum membrane proteins, signals in the cytoplasmic tails of some Golgi proteins mediate the retrieval of these proteins from subsequent compartments along the secretory pathway. The constitutive secretory pathway, which operates in all cells, leads to a continual unregulated protein secretion. However, some cells also possess a distinct regulated secretory pathway in which specific proteins are secreted in response to environmental signals. Examples of regulated secretion include the release of hormones from endocrine cells, the release of neurotransmitters from neurons, and the release of digestive enzymes from the pancreatic acinar cells. The best characterized pathway of protein sorting in the Golgi is the selective transport of proteins to lysosomes. As already discussed, luminal lysosomal proteins are marked by mannose-6 phosphates that are formed by modification of the end-linked oligosaccharides shortly after entry into the Golgi apparatus. In yeasts and plant cells which lack lysosomes, proteins are transported from the Golgi apparatus to an additional destination, the vacuole. Vacuoles assume the functions of lysosomes in these cells as well as performing a variety of other tasks such as the storage of nutrients and the maintenance of turgor pressure and osmotic balance. In contrast to lysosomal targeting, proteins are directed to vacuoles by short peptide sequences instead of carbohydrate markers. Let us now summarize what we have learnt in this session. The endoplasmic reticulum is the first branch point in protein sorting. In mammalian cells, proteins destined for secretion, lysosomes or the plasma membrane, are translated on membrane-bound ribosomes and transferred into the rough endoplasmic reticulum as their translation proceeds. Proteins can be targeted to the endoplasmic reticulum either while the translation is still in progress or following completion of translation in the cytosol. In mammalian cells, most proteins are translocated into the ER while they are being translated on membrane bound ribosomes. The Golgi apparatus functions in protein processing and sorting as well as in the synthesis of lipids and polysaccharides. Proteins are transported from the endoplasmic reticulum to the cis Golgi network. From there, they are transported to the Golgi stack, which represent the site of most metabolic activities of the Golgi apparatus. The Golgi apparatus is the site of synthesis of glycolipids, sphingomyelin and the complex polysaccharides of plant cell walls. Here are a few questions for you to work out. 1. What is the role of ribosomes on endoplasmic reticulum? 2. What is the role of rough endoplasmic reticulum in protein synthesis and post-translational modification of proteins? 3. How are proteins targeted to the endoplasmic reticulum? 4. Explain the structural organization of Golgi complex. 5. Explain the process of protein glycosylation within the Golgi. Here are a few books for your reference. The Cell, a Molecular Approach by Jeffrey M. Cooper, Robert E. Hausman, 3rd edition, ASM Press. Molecular Biology of the Cell by Bruce Alberts, Alexander Johnson, Julian Lewis, Martin Raff, Keith Roberts and Peter Walter, 4th edition, New York, Garland Science, 2002. Biochemistry by Jeremy M. Berg, John L. Timosco and Lubert Stryer, 5th edition, W. H. Freeman, 2002. Here are a few websites for your reference: www.ncbi.nlm.nih.gov/books/nbk9930, www.bscb.org/softcell/ribo. Thank you for watching this session. Let's meet in the next session with another topic. Till then, bye.